start.
Okay. Thank you for inviting me, first of all. Right? And well, it has been a while that I was actually talking at Biocast. The X-Files came in between, unfortunately, right? So we had to do a lot of experiments at X-Files, and then the synchrotron somehow kept up with upgrades, right? So they have now capabilities of these small beams, and they can also interrogate microcrystals, so serial crystallography becomes possible. And of course, I think one of the first beamlines that after the upgrade is the ID29 at the ESRF. And I hope that the APS after the upgrade will perform similarly well than the ID29 at ESRF. And then everybody will show up at Biocast. Biocast will be again oversubscribed. Massively oversubscribed, because there are lots of customers so interested in serial crystallography. And this program KINNTREX is part of a solution to analyze these time-resolved data, a large number of time-resolved data that can be collected with serial crystallography in a relatively straightforward way, and analyzed also in a relatively straightforward way. And so I want to give you some overview over the problems that we had with this analysis to extract intermediates and kinetics, and will also then suggest a solution, this program KINNTREX, and that was developed in my lab in the last couple of years. And one of Gabi Bina was the major developer, and it's interesting that he is not around, but maybe I might not see all the participants here now. Anyway, so the motivation is extract intermediates and kinetic mechanisms from time-resolved X-ray data, basically from time series of time-resolved X-ray data, right? That can be whatever time series it could be, collected at the synchrotron, collected at the free electron lasers. But there are remaining challenges with existing software solutions. So the SVD4TX program is 23 years old. It was functioning already in 2001. It was in Fortran, and we need to do something more user-friendly that has a nice GUI and stuff. That we need to do some push button solution, and we have to implement an expert system that hides the inner mechanics of that program from the user, that the user does not need to know what the mathematical details are. So a solution to this is given by machine learning and neural networks, right? So if you already feel that an intermediate is basically a pattern, and neural networks specialize in pattern recognition, right? And one might even make human-like robots, and their behavior is hard to distinguish from reality. So why should not neural network not be able to handle SFX data or whatever else time-resolved X-ray data, right? So I had that feeling that is possible, and we developed here some software as a humble start, and that gives us numerous ways to evolve that software to something really interesting. So let's start with the previous stuff, the SVD for time-resolved crystallography, right? SVDs, it's somewhat a monolithic or singular program that hasn't been replicated by some other group, because I don't know, maybe the complexity is too much, or maybe nobody wants to do it. So we have the SVD4TX does a global analysis of time-resolved crystallographic data. The question is, what is global and what kind of data, right? So, and then what it also does, it extracts intermediates from time-resolved X-ray data. What does that mean, right? How to determine structures of intermediates, right? and it extracts the compatible chemical kinetic mechanisms from the TRX data. So the question is, what is a chemical kinetic mechanism, and what are the problems associated with this, and how many degrees of freedom you have in the analysis, and how many observables you have. And so far, the SVD is a program that can do all these green things, right? And that was, it's already, I mean, was published 21 years ago. So why do we want to have a time-observed experiment, right? So I can show this with an example with a photoactive yellow protein that is a blue light receptor. Everybody knows that, and it shows a photocycle after light absorption with many intermediates here, multiple intermediates on various timescales. And of course, you'd want to do a time-observed experiment to extract the structures of these intermediates, the atomic structures of the intermediates, of these reaction intermediates, as well as the kinetic mechanism that connects these intermediates, right? And that is the essence of a time-observed experiment. This is why we do a time-observed experiment. There's no other reason to make one, a time-observed crystallographic experiment, right? And of course, the reaction is started by somehow light absorption and a trans-cis isomerization. That was investigated already a lot, and you probably know all of this. So, and for PYP, we need, of course, prime probe experiments. We start the reaction with the laser and probe the reaction products with an X-ray beam, right? So the structures of the reaction products with an X-ray beam. So we can vary the time delay, and we get a time series of difference electron, time series of X-ray data. So what can we expect from a time series of X-ray data? And this scheme makes it absolutely clear what we can expect. You see here an energy landscape. Here, this is the energy landscape of the dark state, right? And then you extract, you excite the molecule, and you get an excited state. And the excited state decays through several intermediates back to the dark reference state if you have a cyclic reaction. These intermediates are separated by barriers of activation, and that is what we want to have an idea how these intermediates here, these minima, look like in a time-observed experiment. What can you measure here? Obviously, you cannot measure anything, because you have no molecules. But that already shows you the energy landscape is time independent. It exists regardless whether molecules are in there or not. In order to measure the energy landscape, you need to fill it up with molecules. The easiest thing is to fill up the dark state with molecules. You crystallize the dark state, and then you can determine the structure of the dark state from that crystal. When you initiate, you transfer an ensemble of molecules from the dark state to the excited state here, right? So you could also measure the property of the excited state, and we have done that. But then the excited state decays to the next state, and what you have here is a mixture of these two states that you measure, right? You cannot measure a pure state. You measure the mixture of states which are populated by molecules. And note there is one little molecule that sits on top of the activation energy barrier, but since it's only one molecule and the rest is hundreds of molecules, the occupancy of the energy on top of the energy barrier is minute. It's negligible. You cannot measure anything on top of the energy barrier. You only can measure the molecules in the minima. In the third, and then when time progresses, you get even more mixtures, right? And you measure a mixture of all these molecules until at the end you get back to the reference state, right? Or maybe there's some molecules don't even want to react anymore, right? So this is what you expect, and from this you want to determine the structures of these intermediate states. You have no chance to determine the structure of the transition state. You have no chance to determine an trajectory between this state and that state. You only measure population changes, right? Here, in these intermediates. So the problems are, of course, you want to unmix the mixture, and you want to do the time, you want to determine the time independent structures of the intermediates. Basically, the structure of this energy minimum. This is what you want, and then this of course doesn't change. And then from time dependent data, where the occupation changes, right? The occupancy changes. So from that, we want to also determine the reaction rate coefficient of a mechanism, and we can also vary more parameters, maybe the temperature, and so on, to get more observables to solve some degeneracies in the mechanism. So this requires this global analysis, right? So in order to understand what we're going to do now, we have to generate TRX data, simulate TRX data, and we know that, how to do it. What we don't know how to extract all of this above from the TRX data, so the time-independent structures, the mechanism, and so on. So that's a typically inverse problem, like structure from crystal diffraction is also an inverse problem. We know exactly how to calculate diffraction when we know the structure, but the inverse we don't know, or single particle images, right? So make a structure from single particle images, right? So neural networks are masters of inverse problems, and that makes it interesting. So let's do the forward direction. If you understand the forward direction, you get a feeling for the backward direction. We simulate time-resolved X-ray data. So the first thing is we need to have a general mechanism. We don't care how that general mechanism looks like, but all intermediates have to be connected with each other. Note that we just neglected that part from I1 to the dark state, right? So after light activation. So we have three intermediates that are connected to each other by rate coefficients. And in order to simulate data, we take one candidate mechanism out of this one, or maybe another one that goes from here to there and then back to the ground state. So there are two candidates here from which we can simulate data. Once we have simulated data, we try to analyze the simulated data with a general mechanism and see what comes out, right? So if we're still in the forward direction, we solve the coupled differential equation of the selected mechanism, this one, for example, and you get concentration profiles of the intermediates, right? Concentrations as a function of time, time-dependent concentrations. This is important for these three intermediates. And of course, that goes back to the ground state. What you also see here, that intermediate 3 massively overlaps with intermediate 2, so there might be mixtures of intermediate 2 and intermediate 3 in this time range. So, and then we could combine the time-dependent concentrations here with the time-independent difference maps of the intermediates, right? So we can have difference maps for each intermediate, and you know how to calculate that. This is just the difference between the intermediate structure and the reference structure in the dark, and that gives you some difference electron density. And how to make difference electron density, I'm not even concerned with. You just look at that paper that explains everything, how to make a difference map, right? So you have difference maps, time-independent species difference maps, which are combined with the time-dependent concentrations to get time-dependent difference electron densities. And this is the quantity you measure, right? And this is what you want, right? So that goes then in the inverse direction, right? So you have a lot of difference electron densities which are measured, and on the right side, everything is unknown. The species species difference maps are unknown, and the concentrations are unknown. So you would need to extract from a large number of time-dependent difference electron density maps the concentration profile, which are shown here, and the time-independent difference electron density maps of the pure species. I think you get the point. So then The problem is that the concentration profiles and the time-independent difference electron densities are interrelated. When you get the concentration profiles wrong, the delta, the difference maps are wrong. Or if the difference maps, conversely, if the difference maps are wrong, the concentration profiles are also wrong, right? So if we get nonsensical, chemically nonsensical difference electron density. So the problem is to find a compatible concentration profile in combination with a set of chemically sensible difference maps that satisfy the data that you collected. So this is a compatible concentration profile. That's exactly the one with which the data were simulated. And you see here an incompatible concentration profile that does not account for the mixture between green and yellow here. So any difference electron density that you extract here shows a mixture of I2 and I3. So that's not gonna work. So these are the data, a time series of difference maps that covering this photocycle, right? And the goal is to get observable relaxation rates. You can always observe relaxation rates or relaxation times in a kinetic experiment. And these relaxation times are linear combinations of the rate coefficients. I don't want to go into detail, so you need to understand kinetic mechanisms or kinetics to appreciate this. And then we want to extract difference maps of the intermediates and a compatible chemical kinetic mechanism with rate coefficients that gives us a set of structures, a kinetic mechanism, and realistic rate coefficients. That's also why we conduct the time-observed experiment. We want that information. So how we did this previously with a singular value decomposition, and you will appreciate after I'm done here with the next slides that this is not that easy, right? So that requires some data manipulation, right? So the first thing is we have this time series of difference map, and we can look in a region of interest where the signal is strong and mask this out. We select a region of interest by masking this out. You see that masks only part of the PYP, so that we reduce the number of the volume which we need to analyze. So, and each grid point, and then we vectorize this volume. How do we vectorize this volume? If you remember that a unit cell in a computer is represented on a grid. So each grid point here contains an electron density value or difference electron density value. So you assign the difference electron density value in each grid to an element of a vector. And you can do this for all the entire 3D volume. That goes then from 1 to m, see? 1 to m. So it's an m-dimensional vector, or a vector that lives in m-dimensional space. And then you do this for all your time points. This is here for time point I plus 1, I plus 2, and so on for all your time points. And you get a matrix of these vectors, right? That contain changes in the difference electron density map in this volume. Okay. And then you do an SVD on that. Here's this data matrix. You do an SVD that gives you left singular vectors, exactly as many left singular vectors as you have time points, right? You get singular values here. This is an diagonal matrix, and you get also the right singular vectors, right? Which is which also gives you the number of as many as the number of time points. The good thing is, however, that there are significant singular vectors and insignificant singular vectors. If you have 3 processes, you get 3 significant left singular vectors, and you can look at them because they're nothing but difference maps. So you can distinguish between significant and insignificant left singular vectors. And I show you also that this is related to the number of relaxation rates that you can fit to the right singular vectors. And then you need to analyze the singular value decomposition with the kinetic mechanism. That works by, okay, I find 3 significant left and right singular vectors. I set up a general mechanism, and I select one of the one candidate mechanism from the general mechanism, calculate concentrations, and fit to the right singular vectors. How does that look like? Here, this is the concentration profile that you get from a candidate mechanism. And you need now, and these are the right singular vectors. Here, they're in black, the black dots here. These are right singular vectors. And the squares here are also, is the second right singular vector, and that's the third. And you need to fit them with these concentration profiles. And the only way to do this is to find a scale factor that scales this concentration here, this concentration scales to here, and this concentration scales to there. And you have to take the same concentration profile and fit the second right singular vector, and so on. You globally fit the right singular vectors with the concentration profile. And then you get a number of scale factors here, M1 to M3, this same set, similar, no, each for the different right singular vectors. And you can use these scaling factors to project the left singular vectors onto the intermediate states. So you see, you get 3 intermediate states, which are admixture free if you do it right, and so on, right? That is the SVD in a nutshell, and that gives a lot of problems because the people don't understand what they're doing, right? And they also don't know how to do this projection and so on. I show you an old example that was done by Sudhara Jagopal in 2004 on a mutant of the PYP, and you see here the right singular vectors, which contain the kinetics. And you see already the relaxation times, which can be easily distinguished here. What is done here, it's fitted with 5 exponentials, with a sum of 5 exponentials globally. And this fit of 5 exponentials gives you the relaxation times and gives you also the number of processes that you can expect in this reaction. And then you take this mechanism as a candidate because you have 5 relaxation times, you have 5 processes, you have 5 intermediates, 1, 2, 3, 4, 5. Set up a candidate mechanism with 5 intermediates, calculate the concentrations and fit the concentrations again to the left, the right singular vectors, and then you can do the projection with these scale factors. And you get here a difference a time-independent, mixture-free difference electron density of intermediate 1, 2, and then for this later one, I just showed the later one. And you see clear differences in the structural relaxations between the first one and the later one, right? So there are these 5 intermediates that you see, which could be structurally then characterized by using the difference maps. I don't even want to go into detail how to determine structure from difference maps. Again, you need to read this paper from me in 2023 in structural dynamics. So what you see here, first of all, the relationship between computed concentrations and the RSVs, the scale is lost. So there is no relationship between that and the real concentrations anymore, because you have these scale factors M, right? So that means that all candidate mechanisms of the general mechanism fit equally well to the RSVs. So why do you actually bother? So, and the magnitudes of the observed difference electron density maps are all ignored, right? So because you do this fitting. So you have a loss of information. So that necessitates the reanalysis on the absolute scale using the extracted intermediates and the measured difference electron density maps. That means that you have to generate a a time series of difference electron densities from the intermediates and the concentrations that you were calculating, and compare that to the measured difference electron density maps. And if they don't fit, you need another kinetic mechanism. This is such a pain, right? So we had to have another program called GetMech, another Fortran program that requires another input and another solution of the coupled differential equations. That was such a pain, and it really exceeded the knowledge of many people. So that was at the end. Although this works, right? It was an intellectual problem. It was a nice intellectual problem that we solved, but only a few people ever used it. And that makes it very difficult. So maybe the neural networks are a revelation, right? And so the neural networks will cook everything down to nothing almost, right? So you feed some data and get out some intermediates and a compatible chemical kinetic mechanism. And you have seen how difficult this was with the SVD. Maybe with the neural network, everything is not that complicated, right? So neural networks consist of layers of neurons and corresponding activation functions. And you will see that later, that how to implement that in a computer. So there are popular neural networks like AlphaFold and ChatGTP, GTP or GPT, GTP, right? They do not, but they don't extract difference electron densities and mechanisms, right? So you can not send some difference maps to AlphaFold and tell them get me my intermediates, right? That will not work, right? But we have to do our own neural network. The problem is that neural networks are usually trained. Training is required. But here we don't have training data, nothing. We can't train a neural network that at the end something like a mechanism falls out. This might be an unsurmountable problem because there are no training available. But physics informed neural networks. We can inform neural networks by physics. And they were used by Peter Schwander here in the department for cryo-M data, right? They don't need training data. And the physics-informed network extracts parameter and data pattern that satisfies the relevant equation with which it is inspired or informed. That's the important thing. So, and the neural networks can work directly with raw data or any other data you provide, but you need to inform it by physics. In our case, we have to inform it by a kinetics. So that's why it's called KINNTREX, kinetics informed neural networks for neural network for time-resolved X-ray crystallography. It's a neural network that we need to inform by a chemical kinetic mechanism. Therefore, the output will be mechanism-like. If we would inform it by diffusion, then the output would be diffusion-like. But here, the output will be mechanism-like. And it is a neural network that is also informed by the result of a singular value decomposition. So the output will also be projection-like. And that's what we want, right? So there, of course, we can immediately put in some restraints on the rate coefficient of the mechanism. So the rate coefficients cannot be larger than the largest observed relaxation rate. If we see a relaxation rate of 10 to the 8 per second, the rate coefficient cannot be 10 to the 10 per second, because that would violate the largest observed relaxation rate. So we cannot use that. So we have a restriction by the largest observed relaxation rate. So for an upper limit of the rate coefficients. And then the lower limit is of course 0, right? So rate coefficient must be positive. So then the next restriction is, of course, the number of intermediates must be equal to the number of relaxation rates or the number of significant singular vectors and values, right? That's another restriction, powerful, that we can put in to physically restrict that network. So the goals, of course, you need to remember, we want to get the time-independent difference electron densities and a chemical kinetic mechanism, right? So with the intermediates and rate coefficients. And that from a time series of time-dependent difference electron density maps, right? Here is then our KINNTREX, right? Our neural network. This is a little bit an overview that shows everything. Here, this is the data matrix, which I told you before that we were generating by vectorizing the region of interest. So we stuff them into an SVD, and the SVD says us, oh, we have three processes going on. So we can already say, oh, three processes, great. We restrict our network to these three processes. And then the SVD will also give us an idea about the largest relaxation rate. So we will restrict all relaxation rates of our mechanism to the largest and to zero, right? So, and then there are two neural networks, a projection neural network that does the projection, and you have struggled probably very much to understand what the projection does. The projection neural network does it for you automatically. Then there's the conversion network that generates concentrations, or that generates rate coefficients from concentrations. Then we hear this is the part that, with which we inform the neural network. This is basically the couple differential equations of the mechanism. And then we calculate the loss function and do a back propagation and do this 100,000 times. Right. So, and then after 100,000 times, we hope that this is settling, and out comes the intermediates, the rate coefficients, and the loss value, basically how good the fit is between the predicted data and the input data, right? So let's start with the projection network, with this one, right? So the input layer here, these are the left singular vectors, right? 1, 2, 3 to m, right? So the significant left singular vectors. And then the middle layer consists of the time-independent difference maps of the intermediates. Oh, that is already what we want, right? And on the output layer contains the calculated electron density, difference electron density maps that can be compared to the experimental difference electron density maps. And of course, these are connected by a matrix MM, and the matrix MM, if you have three processes, it's a 3x3 matrix, right? So therefore, it's a partly connected feed-forward neural network. And the CNN, this is a matrix that contains weights that relates to concentrations, and AMM is the projection matrix that generates the intermediates from the left singular vectors. Okay, so that's a square matrix, and CNN is an MP matrix. M is the number of voxels that you have in your region of interest, and P is the number of time points. So that gives us automatically difference maps and intermediates, time-dependent difference maps and time-independent difference maps of an intermediates. But when you start with this, these weights are randomly initiated. So this I and the E doesn't make sense at all. There's only garbage in there. So then we forward the weights here, the forwards after rectifying, we forward these weights to the so-called conversion network. And we will talk about conversion network in the next slide. Here, the conversion neural network. It's a fully connected network with the middle layer, with one layer. So it's not deep learning, it's shallow learning, because you have only one layer here, one layer with neurons in the middle. And the input are concentrations of the intermediates, and the output are rate coefficients. This is crazy to determine. You have first rate concentrations, and you want to make rate coefficients from the concentrations. It's usually the other way around. You have rate coefficients and you make concentrations. But you see this inverse problem. Yeah. Here you have concentrations and you make rate coefficients out of it. So there you have two matrices, the weight matrix W1 and the weight matrix W2. They connect the input and the output layers. So, and these become meaningful after sufficient information accumulates. These are also initiated with random numbers and generate garbage case. So then the next part is the kinetics information. So the kinetics information takes these Ks, puts them in a coefficient matrix, in a model matrix K, and then the model matrix K is diagonalized, and you get concentrations CDE. So you see you have two places where you get concentrations, CNN here and CDE here, right? These CDEs comes after diagonalization of the coefficient matrix, and the CNN come from the projection network. And at the end of the day, you can calculate with the intermediates that you also get from the projection network and these concentrations, a set of time-dependent difference maps, which can be compared to the input difference maps, right? So, and that gives us a handle on that. So, because we have a loss function, and that loss function is nothing but the mean square displacement or mean square difference between the experimental between the difference electron densities EC2, which are generated here, and the experimental difference electron densities. You have you can also compare the concentrations that derived here, CDE, with the concentrations which are predicted here in the conversion network. And you can calculate also a difference between the EC1 and also this the experimental difference electron density maps. And that goes all in the loss function, and you want to make the difference as small as possible. How do you do this? You do this with the so-called back propagation. You update the weights AMM, CM, CNN, and as well as W1 and W2 until, and you iterate until the loss function is minimized. So but you see here also the BL and the BH. The BL and the BH these are the lower limits and the higher limits of the rate coefficients. If they exceed the BH or are below the BL, then the loss function will also increase. So the loss function forces the limits of the rate coefficients to be within BL and BH. So let's look a little bit at the size of these matrices. I hope you're not lost. You can imagine that, of course, you have in the region of interest, you have roughly 2,290 or so grid points, right? That gives you your the size of a left singular vector here. That is the dimensionality of the space which you're working on. And you have 21 time points, right? Well, N is the size of the region of interest, and you have 21 time points. That is all in the experimentally measured difference maps. Of course, you select only 3 left singular vectors. Therefore, you have the same number of grid points, but only 3 significant left singular vectors. That means that the matrix MMM is 3 by 3. Of course, the intermediates have this have the same region of interest than that extracted from the data, right? Also 2,901 at 2,291 grid points, but we have only 3 intermediates. So you see that this is connected here. And then the concentrations, the weight matrix, which is here, of course, has 3 for the number of intermediates and 21 time points. So you can reconstruct basically from the intermediates and the concentrations, you can reconstruct a set of difference maps which are here, the EC1 that can be compared to the measured difference maps. So then you transfer the CNN into the conversion network, and the CNN will be linear, will be flattened. Basically, the CNN, the rows will be concatenated. So you get a vector that has a 63, those 3 times 21 rows basically in one column. So you get a column vector with 63 entries. And then it turns out that, of course, these matrices W1 has to be the same, 63, but, and then 25. 25 has to be chosen so that the number here is larger than the number of rate coefficients. And we have here 10, right? So W2 will then also show that we have 10 rate coefficients. So that the matrix has to be 10 by 25. And at the end, you get the vector of rate coefficients with 10 entries, right? So, and that all of this has been implemented by Gabi Bina in our lab with Python and PyTorch, right? So all matrices are tensors, and these functions that you need, the rectifier and the backpropagation minimizer, are all implemented already in PyTorch. That's why PyTorch exists, right? So the model matrix K and the coupled differential equation solver was implemented by Gabi, also with the help of Peter. And so we got some prototype software, and we had to test that, of course, with simulated data, right? All data and information is in a single ini file, and the results will be stored periodically. Once the computer breaks down, we can start from there. The program runs on CPUs, and it's not parallel, right? But multiple instances of that program can be run on multiple processors if needed. So 100,000 iterations takes maybe an hour on a fast PC, and 100 instances to check for similar output can be run on a cluster with 100 CPUs, and also requires 1 hour. So you will see why we have to run 100 instances, right, to check for similar output in a minute. So what are the results? You have the ground truth data, which are shown here. These are the time-independent difference electron densities with which we were calculated simulated the time-dependent difference electron densities. And then we, from a general mechanism, which is shown here, we selected a sequential mechanism, a candidate, and a dead-end mechanism for the simulations. And you see that they generate quite different concentrations. So in the time-dependent difference maps were, as you see, at 21 time points, right, for an imaginary PYP photocycle, by combining the time-independent difference electron density maps with the time-dependent concentrations calculated from that mechanism, right? We apply experimental noise so that the data are noisy. We mask out that volume here that gives us these 2,000 or so grid points. And then KINNTREX predicts 3 left singular vectors, and we use then 3 processes. And we produce and we analyze those data that are produced like this with a general kinetic mechanism, this one, where nothing is known about it, except the number of intermediates and some restraints for the rate coefficients. So let's try with these are the simulated data, right? And that was these simulated data were subjected to KINNTREX, and nothing was known except the number of intermediates and the range of rate coefficients. And you see, most of the rate coefficients go straight down to 0, and 3 rate coefficients survive. And the number and the numerical value of the rate coefficients are exactly those with which we simulated the data. This is shown here by the dashed lines, right? So after, say, 100,000 iterations. In this case, this is done, right? And so everything converges. And here you see also the comparison between the ground truth, which are these spheres here, and the calculated, the extracted concentrations, predicted concentrations. And also down here, you see the different selectron density maps that were extracted of the intermediates, which match exactly those which we have used for the simulation, for the ground truth. So that's very encouraging. But now we do this for that mechanism that's more complicated because it has this super strong admixture right here. So also after maybe even 30 or 40,000 iterations, everything converges. In most of the rate coefficients went to 0. Only a few were meaningful. And from that, you can calculate these concentrations. And they fit acceptably to the ground truth. And they're also acceptably extracted difference electron density map. And you see that this difference electron density map and that difference electron density map have no mixtures. Otherwise, you would see a little bit of that in here and a little bit of that in here. So these difference maps are really mixture free. So we have separated this strong admixtures, which would usually quite difficult to achieve. So in order to look how good this works, we had to repeat this experiment 100 times. And since we have the ground truth data, we can compare the concentrations that were spit out to the ground truth concentrations and make a histogram. Each time we get some new concentrations, we calculate this factor RW, basically a figure of merit, how often you find of the difference between the concentrations. And the histograms are extremely sharp, right? They peak at extremely sharp values. That means that each and every time the correct concentrations were extracted. And if there is a deviation, it's very small. But there are larger deviations, of course, but they can be easily distinguished. And then the rate coefficients from these histograms can be then extracted. But we don't have the ground truth. You understand that in a real experiment, we don't have the ground truth, right? So what can we do there? Well, we can look at the rate coefficients themselves and look how they vary when you run the program 100 times. And here are histograms of rate coefficients extracted by analyzing these kind of scenarios here. And this is also a very difficult case with a lot of overlap, but you still get sharp histograms for the rate coefficients. So that means that the rate coefficients can be reproducibly determined by the program. Here also very sharp rate coefficients, and the dashed lines are always the ground truth, which of course in a real experiment you don't know. But here we can show them in this graphics in order to show how good the rate coefficients are predicted. And then here these two blue histograms basically show the same rate coefficient once here and there and there, and that is a degenerate mechanism that looks exactly the same, except that the order of intermediate 1 and intermediate 2 is reversed, so that nobody cares about that, right? So but in general, the extraction works quite nicely and is very reproducible. So we can say that the approach using KINNTREX works, and the appropriate concentration profiles are predicted, and the mechanism can be degenerate as long as they produce the same concentrations, because only if we have compatible concentrations, we will get compatible difference electron densities of the intermediates, which are admixture free. KINNTREX is very stable in contrast to SVD40X, where you always have to be careful for the fitting, right? And GetMac is not required because you do the analysis, the comparison directly if you go back here, oh God, where's my KINNTREX? Here, because you can do the comparison here directly on the difference electron density level. You compare the measured difference electron densities directly with the predicted difference electron densities, and you do not have to go through the right singular vectors and do a fit. So user input is also at minimum. The only thing that the KINNTREX needs to know how many processes are there, and the minimum and maximum rate curve, the range of rate coefficients that are in the data, and it's a clear potential for push button operation. So far, we have implemented only the single step mechanisms, right? So one intermediate goes into the next and into the next. But there are also other mechanisms, especially when you look at enzymes. So there are usually second order rate coefficients. That's that characterizes the binding of a ligand, for example. That is the second order rate coefficient. So that elegant model of matrix diagonalization does probably not work, and we have to find a way to also implement that. So, but what is very important for me, because 10 years ago, I think we were all excited in order to change the temperature and get some barriers of activation from time resolved X-ray data by not by 5-dimensional time resolved X-ray data. We varied the temperature, we extracted barriers of activation, and that was also not that easy. That was not very straightforward. But if we would inform the network by the Boltzmann equation, right, then we would immediately be able to analyze the 5D data. It only requires one step more, and we would feed all 5D data. So in this case, at our case, that was 14 temperature points, and 30 at each temperature, there were on the order of 30 difference maps into that program, and it would extract not only the intermediates, hopefully, that are compatible, but also use the temperature as an additional observables to tie down or to remove the degeneracy of these mechanisms, which I have told you before, that exist when we have only a single time series at a single temperature. So that gives us finally a handle to do this elegantly and user-friendly, so that people can do 5-dimensional crystallography in the future without having problems with running the programs. So what the future is of KINNTREX is, of course, an intuitive graphic user interface, right? We need to apply this to real data, and we have to apply this to 5-dimensional X-ray data, and maybe additional dimensions, like you can do pH and so on, right? And then it will be, of course, more difficult to have a physical connection between the structural changes and the pH, for example. Then it's difficult to find a physical information that we can implement. So what also needs to be implemented is unit cell reconstruction, right? That SVD did that. The SVD for TX program did that. That incorporates the non-crystallographic symmetry and potential unit cell by crystallographic symmetry. And then we have still non-crystallographic symmetry. So for example, in an enzyme like this that has 4 subunits, we have fourfold non-crystallographic symmetry, and each subunit here reacts different from the other subunit. So that's not that easy. So we have to incorporate that. And because we want to extract the kinetic differences in between the subunits. The next step would be to get extrapolated electron density to model intermediates, because you cannot take difference electron densities to model structures of intermediates. So we need to have an extrapolated electron density. So there's a lot of thought that went into it, to how to make them. And then finally, we can make frames for this kind of movies. This is not working with intermediates and kinetic mechanisms. Maybe it works like that. Yeah, fantastic. No. Pointer options. Now it works. So we should be able at the end to make movies that took forever to make here for this enzyme with another SVD analysis by simply providing the data to the neural network and get something like, you know, binding of ligand and reaction of ligand with the protein from the SVD data alone, from the neural network alone. Here, appearance of substrate and the reaction of the substrate with the active center of that enzyme, which is of course an extrapolated movie, and it's not really clear whether that works like this. It's a guide to the eye and a fantasy. The movies extracted by KINNTREX would look, of course, absolutely different. They would only show changes in population. So the far future is KINTREAM. KINTREAM would analyze time-resolved cryo-EM data, right? So first of all, the problem with time-resolved cryo-EM that nobody can sort the data into intermediates in the moment, right? So that would be done by the network, right? And then determining the dynamics of the entire particle ensemble, and that gives an additional level of complication, right? It sounds too good to be true that this is working. We need fantastic programmers. Gabi was a fantastic programmer, but unfortunately the BioXFL funding ran out. So Gabi had to go to Peter Schwander now, does his work there. And I would have loved to retain him to develop further properties of KINNTREX, right? So I have to wait until the NSF or NIH is willing to provide some funding for the further development. And here are the people who are involved. Gabi, of course, who did the programming work, and the tech, who was also advising on singular value decomposition, and Peter, who basically provided the expertise for a lot of details of this program, right? And the funding, of course, is from the NSF, and everything is published in IOCR Journal 11 in last month, or this month, actually. So I thank you also for your attention.
Hello again, this is Irina Kashlova. I thank you so much, Marius and Gabi Tech and Peter, for working and sorting out this complicated application, really complicated subject, at least for me. Does anybody have questions? It's 11, but perhaps we'll have time for a few questions. Yes, please. I saw a hand, but I missed who. Please unmute yourself and say your name and ask your question. Thank you.
Yeah. Hi, Michael. Or sorry, Marius, Professor Schmidt. That was a great presentation, and thanks also, Gabi, for being on the call. This is Arnold. I'm a student at Northwestern University, and I don't do crystallography work, but we use similar kinetic modeling in our time-resolved SACS experiments. I'd love to talk about the model more, and so it really does seem like a benefit to use the neural network in the sense that you don't have to provide a kinetic model. So the benefit, in my understanding, would be that, like normally, I would just sit down and look at the number of intermediates, map out, the different kinetic states, and just, do some sort of fitting, right? Still using SVD. like 3 times, right? And so the benefit here I wouldn't need to do that 3 times, and the benefit would just be that I would just run this once and it would provide me a model. Is that correct?
It's exactly correct. You
Okay.
only have to do the right information, give the right information to the network. The network will not extract anything that is not by which it is not informed. It will be the model will be part of the general information that you give it. If you give them a diffusion, then it will generate a model based on diffusion. If you give it a kinetic mechanism, it will generate something based on a kinetic mechanism, right? That's the
Understood.
constraint.
Okay, then a quick follow-up to this is, I don't want to be overly critical, but my understanding of, deep learning and networks is often they're used for classification problems more so than regression problems.
Exactly.
And the way that we do fitting and optimization now is using like a least squares based method, right? Gauss-Newton method to invert matrices over and over to get a really global minima. And like sometimes neural networks are criticized for regression because they use, gradient descent and it just jumps to the nearest local minima. Is there a way to account for this in your modeling, like in your studies? Like how do you comment on the uniqueness of a solution given that things like Newton's method is the established way of solving inverse problems?
Yeah. Here, of course, you have seen that we have to repeat this 100 times and we get this narrow distributions, this narrow histograms. And this is these histograms don't we have of course initiated the network with a range of different weights, right? So it is independent how you initiate the weights, so they're all converge to the same set of values, basically, right?
Okay.
100 times, so it's very reproducible. You get this very narrow histogram, right? And that can be also done with data where you don't know the ground truth. You repeat this 100 times in an hour, basically, with 100 CPUs, and you get narrow histograms. And in the moment you get narrow histograms, your solution is most likely right.
I see. Thanks. I appreciate that.
I have also something about the optimization. You don't have to use gradient descent, right? Stochastic gradient descent. You can use different optimization problems. Torch has multiple of these options, so just use whichever better fits you.
Okay, thank you.
Yeah, but I also have to say this is like the SVD program that did a lot of fitting, and this one is also nothing but a real glorified, really, I think, an elegant and intelligent way to fit some data with the model.
Okay. Next question coming from Vatsal Purohit. I'm sorry if I misspell your name. Yeah. Vatsal, could you please just your question? Sure.
Hi, I'm actually going to ask the question. Hi, I'm Sahand. I'm a graduate student at Emory University, where we work in time-resolved crystallography. We've only ever used SVD on spectroscopic studies, but I was wondering, can you use target testing? I'm sure you're familiar.
No, I don't know. What is target testing?
So target testing, at least in spectroscopy, you're able to give it a state that you think is present and use your left singular values in order to see if that state is present within the mechanism.
Yeah, well, certainly if we have enough time, we can do this. But at the moment, we do not do any such thing as target testing.
Okay,
would be of course cool. I mean, in this case, we know the target. It's the ground truth, and we tested this, and there's always the ground truth that falls out, right? So,
sorry, the target is
the ground truth, right? So we have the ground truth. That's our target, and we just start with anything random and end up with the target, and that's all what we do here, right? When we looked whether that program works.
So the target could be any of the intermediate states. as well. So I guess, I mean, if it's not something you pursued, I guess I was just wondering that, but also it might be able to better inform the neural network, because if you have a proposed intermediate and it doesn't show up as a target, then it wouldn't be able to. But sorry, I'm
Okay.
That's totally possible, I think.
Okay. Okay.
Okay, can I ask one question? This is Irina. Marius, this work, do you have it on the GitHub, or how the code is available, or this is just available for your lab and you would expand later?
Yeah, we have stored it in a local ice cream store, but
Right,
Gabi
right.
can say more about that.
Yeah, so it's actually on the GitHub. I can try to, how do I write down the
in the chat?
Perhaps in the chat. There is this chat on the bottom.
Yeah, I can write
And
add
you
that.
can write in the chat, yeah. But
Okay, I'll do that right now. So
yeah, we also doing SACs here. Would this program work without modification if we will using 1D data instead of three-dimensional? It should be, right?
yeah, I think so.
The good thing is that the SACS data are already vectorized. So you simply take the vector of the spectrum and you don't have to do nothing, right? So I think that should also work right away. If you provide a mechanism, inform this thing with the mechanism, it will pull out from the SACS data directly intermediate data, intermediate SACS-VAX spectra. Right?
Yeah, I'm waiting. But again, you know, if I don't see anything in the chat yet.
Oh,
No,
But,
I'm adding it right now.
okay. But we can find it like KINNTREX and GitHub, and probably we can find this link. Duque?
Hi, this is Duca Hekstra from Harvard University. I thought it was very inspiring, and I was wondering about two things. One is if you could do this directly on amplitudes instead of on electron density.
Okay. I'm not a fan of amplitudes, I must say, right? Because the amplitudes basically the signal is distributed over all amplitudes in reciprocal space, right?
Right.
And if you use the phases of the dark state and the difference Fourier approximation, you concentrate the signal very much to a region of interest, right? And then the signal to noise in the region of interest is much better than what you expect from amplitudes, right? So, but since the amplitudes are measured very accurately nowadays, I think one can also try to extract simply from amplitudes the same thing, or from difference amplitudes. I would not even say amplitudes. I would take difference amplitudes, right? And then we get a set of difference amplitudes where one can also try to do some difference electron densities and look whether they're the same as we get when we calculate difference electron density first and then extract, right? That would be also a consistency check, at least for that.
Right. And I was wondering, is it possible to use at least these kind of simulations as a way to improve your experimental design in terms of how many time points you need, how you need to space them?
I don't think so. The thing is that we never know when the intermediates peak,
right?
And we never know at which time scale they appear. So the experiment would be a random experiment where we have to take care of the fast and slow process at the same time. And I haven't come up with anything better than logarithmic time scale, which weighs the lowest and the fastest time scales in the same way, right? So.
All right, thank you.
Okay. This is 12:11, and I believe if there is no raised hand, then we can conclude our seminar. Marius and Gabi, Tech, and Peter, thank you for wonderful work, very inspiring. Again, if anybody would like to present, and Marius, thank you so much for making this complicated subject a little bit less complicated in your presentation. Now, yeah, I would like to tell if anybody would like to present your work or find some subject interesting, please send us your suggestions. We will be happy to host you or consider suggestions. Also, I would like to tell that many of you reached asking what is Biker's plans in terms of upgrade, and Rob will be talking about it in the beginning of June, I believe, in a couple of weeks. And we know that this restart of the operations a little bit delayed, like maybe 6 weeks as it is for now. So it will give everybody enough time. Thank you so much for everybody for coming. Thank you so much, Marius. Have a good day now. Goodbye.